Can't remove the mask. Okay, thank you.
Just waiting for everything to be set up. Okay.
Bueno, pues buenos días y gracias por venir a este nuevo seminario, segundo con el ciclo de seminarios que tenemos, especialmente a la gente de Santiago que se ha acercado desde aquí. Y él es Sándor Gonda, que es profesor de la Universidad de Debrecen, que está en el este de Hungría, y va a hacer una presentación acerca de él, que me dijo que ya tienes algunas slides, así que él mismo se va a presentar y nos va a contar un poco el trabajo que está haciendo sobre la relación entre hongos endófitos y una especie de Brassicaceae, que es horseradish, y bueno, es un poco un trabajo en común que tiene con nuestro grupo, entonces estamos estos días aquí buscando posibles colaboraciones. So, Sándor.
Thank you very much. I am Sándor Gonda. Sándor is the Hungarian equivalent of Alejandro, something like that. Gonda means Gonda, and I came from Debrecen, that's the eastern part of Hungary, and I would like to talk about this chemistry and fungal community correlations and some of our recent results. First, the acknowledgements. I am very happy to be here. Thank Pablo for helping me putting this all together. And I thank all of you for coming, or listening. Of course, this work would have not been possible with family, colleagues, partners, financial support, and those who develop the software that we use. So this is a collaborative work. Debrecen is in the eastern part of the Hungary. It's the same time zone as Pontevedra, but other things 2 hours apart. So it's very interesting to see that the sun rises and sets completely different time. City was mentioned quite a few centuries ago. It currently has a population around 200,000, and this is still true. So it's a guy who went there at the end of the 18th century said, I don't know about the people even lived there. There's no river, no spring, no nothing, no wood, no construction material, no good wine. But we managed to stay there anyways. This is my home university. This is one of the main buildings. It is a merger of several universities of Debrecen. So there was a separate medical university, a separate agricultural university before 2000. It merged and became this large, complex university with lots of campuses, almost 30,000 students, a lot of international students, and more than 1,500 of academic staff. So that's who we are. And I am a researcher at the Department of Botany that also has lots of research groups. And I came from the ones who are primarily interested in the chemistry of living organisms. Okay, the roadmap. This is what I'm going to talk about. First, why do metabolomics instead of quantitative chemical analysis? Why complement quantitative chemical analysis with metabolomics if you have the opportunity? Can you measure the adaptation of the endophytic fungi to this chemical environment, the plant, inside the plant? Is it possible? And how does it work out? Is this measurable outside in the field? Okay, so let's start with metabolomics. What is metabolomics anyway? It is part of the omics sciences. That is this row. It's like genomics, transcriptomics, and anything omics. There are a lot of buzzwords that end in omics. And these buzzwords always tell you that you are interested in the layer as a whole, every compound, every protein, every gene, etc. So in this case, we are interested in every chemical compound that are small molecules, okay? We are trying to use this to find correlations, associations with some phenotype, okay? So it's like disease resistance or anything else regarding plants. We have to data mine to get these phenomena. It's not trivial to number crunch that amount of data, so we need some visualization techniques. And it's also a buzzword, but I might say it's interdisciplinary because you use analytical chemistry and bioinformatics to answer scientific or agricultural questions. And of course, you can also use metabolomics for petrochemistry and medical sciences as well. What are the main differences when compared to traditional quantification by, say, HPLC? We use the same instruments in metabolomics. Of course, you have to use hyphenated techniques with mass spectrometry, but the instrument is essentially the same. So what are the differences? In quantitative analysis, you pick a few compounds, you have traditional calibration methods, and you end up having absolute concentrations within your sample. That means it's micrograms per gram, something like that, micromole per gram. We have massive, strict validation protocols for those methods because the pharmaceutical industry required these to be present and it spread all over, okay? If I have a peak that has the same retention times, same spectral characteristics, then I have identified something. On the other hand, with metabolomics, we only have relative abundances. I can say that sample A contains the most among my data set and sample Z contains the least, something like that. Because of that, we do not have any standards, we do not use any authentic standards. We can measure the relative abundance of hundreds, thousands of compounds simultaneously. Validation protocols, however, are a matter of lots of debate now. So no one knows actually. Go check. Of course, just go check. Yeah. Okay. Yeah, is she okay? People are turning. Okay. Yeah.
They're from Santiago, and they came from Santiago.
Sorry.
Last time you came to the talk, I think.
unfortunate.
Yes. Yeah. Okay, you can continue.
Okay,
It's okay.
it's okay. Okay, okay. So, thank you. Okay, so because we have no authentic standards, we are left with iterative identifications of the compounds. I'll explain that when it comes to examples. So we have no authentic standards. That means that we have no prejudicative idea of what compounds would be the most interesting. Therefore, we can find biomarkers. If I discard this opportunity and use untargeted metabolomics, I am going to get this. It's not free. The quality control, so the measures that you have to take to be able to tell that the instrument worked fine during the sequence, you need quality control measures, which means quality control samples of some sort. It's going to increase the measurement time, the instrument time by about a third. It's quite uncomfortable, but that's the way it is done. However, once you started with this approach, later on, it's easy to upgrade to quantification. You pick a few samples of the data set. You already know that your compound of interest is most abundant in sample A and least abundant in sample Z. And you can build some pseudo calibration curves to assess the approximate amount. However, if you started with quantitative analysis, you cannot upgrade to metabolomics. You have to start everything from the very beginning because you have no QCs. QCs, I won't go in detail. These pooled samples, there are 4 or 5 strategies how to generate a QC sample. That could be a separate presentation, but they're injected there with 6-4 injection. We use them for precision, local sensitivity, adjustment for all analytes of interest. We assess linearity, range of determination, ATC, and all these result in filters that help you determine from the raw data which peaks are from your sample and which peaks are likely to be from your sample. It can be run in targeted and untargeted manner. I vote for this one, but it is harder to conclude these results if you do not find anything, which is quite possible. These structures, the plants themselves are complex, so it's you can maximize coverage of for compound polarity and compound biochemical group, but it is not sure that the compound will pass the quality filters. Okay. And there is always going to be a compromise. If you use water and methanol mixtures, which is typical, that's not optimal for lipids. You'll see lipids, some more polar lipids, but not optimal for lipids. Okay. You can do targeted if you know that your plants contain something that you are specifically interested in. You can use specific fragments to check for glucosinolates, for example. And you can do bioinformatics level targeted filters as well. Okay, let's move to the plant. This is horseradish roots. It's a significant product of my region, and we've been using it as a model in these endophyte, metabolite interaction studies for quite a while now. It's also a crop, so it's not only a model. We eat it Easter time. We consume the most, but actually, it's most of it is exported to Germany. Why is this a good model for these studies? It is stuffed with glucosinolates. Glucosinolates are the major chemicals of the Brassicaceae plants, and this plant contains a few percent per dry weight. That's very, very large compared to other species. The major ones are cyanogrin with this alkyl side chain and gluconostatin with this benzenic or aromatic side chain, but it also contains indolic ones and this sulfur-containing versions as well. Our questions were how do the endophytes interact with this biochemical pathway? So these are the precursors of the actual bioactive agents, the ITCs that are against insects, fungi, and bacteria, and nitrates and other miscellaneous things that arise and actually are less understood what they do in the plant because they are not as bioactive, not as toxic. Okay, there are several papers on insect interactions, but the literature is dwarfed by that of the ITCs. So, what do the endophytes do to this whole thing? How do they tolerate ITCs? Can they decompose glucosinolates? Can they produce any of these compounds? These were the questions that we tried to answer. The interest in endophytes stems from a few things. First of all, it's the apparently non-pathogenic nature in plants in general, as well as crops. So it's this biocontrol agent approach is one of the major drivers, and the other one is the possibility, the higher chance to find bioactive compounds if you purify some natural products from the fungus. So these are the perhaps the which led us starting to work with these fungi in the first place. These are a few confocal microscope photos which show that the fungi can well recolonize in vitro plantlet roots of horseradish, so are not just accidentally considered as endophytes. So this is from our set. Our first experiment I want to talk about was very simple. We prepare a horseradish extract that's pretty much rich in glucosinolates, but otherwise it's a total extract. There is plenty of nitrogen-containing nutrients, amino acids, peptides, everything there, and use it as a fungal medium. We add the endophytes, allow it to grow, lyophilize it, reuptake a solvent, and then characterize the decomposition of the glucosinolates with LCSMS. Okay? These numbers E denote the various strains for this first study. C is for control, and we're seeing the massive decomposition of this cyanogrin. We are talking about quite high, a few milligram per milliliter amounts here in the liquid. It turned out that 6 of 7 endophytes are very efficient decomposers. Okay. Apparently, the same was found for the benzenic gluconostatin, the sulfur-containing glucoverin, and the glucobrassicin were a bit more resistant toward this fungal transformation, but anyways, most of it was decomposed. Minor note that there are two pairs strains here. E1 and E3 are from the same species, and E5 and E6 are also from the same species. Look at this within species variability. These are both Fusarium oxysporum strains, and one is much more efficient than the other one. Okay, so the next logical question is: do they produce anything volatile? Do they produce the same compounds as the plant during the decomposition? So we came up with this headspace system. It either contained liquid or agar solidified horseradish extract, or a thin film of the same agar solidified, so that we get a larger surface. Okay. It was inoculated with the fungi. They started generating some volatile compounds, and we measured the pattern with GCMS, headspace GCMS. So we are not using an adsorbent filament here. We're injecting air directly, okay? What could be found is a control, a few endophytic strains, and a few salt fungi as in the upper two chromatograms. But what's here? Allyl cyanide, cyanogrin decomposition product, okay. Allyl isothiocyanate, AITC. Okay, not all of the fungi seem to be active. This is only a subset, but there are specific products like esters, butanol, etc. But there is something wrong with this data set, one might ask. So it's like there are strains that do not produce any significant amount. Under significant, I mean compared to control. So like there is not much strains that generate a meaningful amount of this specific decomposition product. So what's happening here? What is the product of generation? We screened other sulfur-containing compounds, and we found a few, but the whole thing doesn't make much sense at this point. What if isothiocyanates are generated indeed, but they are not detectable by the classical methods that everyone tries to detect them? They are very reactive towards thiols of glutathione, protein side chains of cysteine, ATC. What if the main chemical form to be found is some sort of adduct between the glutathione or other sulfur-containing small peptide? Glutathione is very abundant in fungi. And the AITCs. It forms a dithiocarbamate immediately if they meet under suitable pH conditions. Okay, and checking the extract again with LCMS specifically for this compound, it turns out that it's quite abundant, and it's there in many of the cases. This is a standard that we synthesize the compound. These are the controls. It's practically absent, and we have a lot of fungi that produced it. So the glutathione adduct of AITC seems to be a good candidate to check whether there is a decomposition that you cannot measure by GCMS. It's not volatile anymore. The same fungi, the juice, the medium of the same fungi contained a lot of this. There were also fungi that contained this compound. For example, the Setophroma terrestris strain, this E4. It generated no volatile AITC, but generated this compound, perhaps because it picked up the cyanogrin, it metabolized it intracellularly, and the AITC couldn't even escape. Something like that. Okay. Comparison between endophytes and salt fungi continued with the with the same approach. We used soil fungi from the same soil as the horseradish's control and compared the end-time residual glucosinolate content. This is what we found. From this point to the right, these are soil fungi, and the yellow ones are, the orange ones are endophytes. This is a control, nothing happened. What are we seeing here? We're seeing here that it's like 6 of 7 versus 4.5 versus 10 for cyanide. The amount of the number of fungi is not enough to come up with a statistically significant difference in a Fisher exact test, for example. But this looks like something that's meaningful. Okay? The same goes for gluconostutzin, though looks like this strain, which is the same species as this strain, wasn't that effective. And again, glucoiberine and glucobrassicin were a bit more resistant, but the amount of significant differences compared to control and the extent raises some questions. So it looks like there is some sort of adaptation to the chemical environment on the endophytes compared to the soil fungi. Let's take this a bit further and let us see whether the fungi can use these precursors built against them as nutrients. So we prepare an inorganic medium with no sugar, and the only organic carbon source will be the compound cyanide. Okay? It would be equimolar, so that the same glucose is added. It's quite concentrated, but yeah, we run a plate and measure the absorbance of the mycelium. This case, it's 4 out of 7 against 2 out of 10. So it's a thin difference, but perhaps it's there. 4 fungi accepted the 4 endophytes accepted the cyanide as the sole carbon source. Interestingly, these ones who generated the volatile AITC did not. Perhaps they use an extracellular enzyme, therefore they emitted volatile AITC, but couldn't use this as a carbon source. So they needed sugar to start. The legend is the black bars are cyanide, and we are comparing against water, which is the white, and the gray, which is the sugar reference. Okay. Interestingly, there are also fungi who cannot grow on inorganic nitrogen, but soil is diverse, soil communities are diverse, so it's perhaps not a surprise. Anyway, a thin difference here as well. Another viewpoint of the same story is a thiocyanate tolerance. Do the endophytes manage to deal with isothiocyanates better than a set of random fungi? Liquid culture, IC50 determination, lots of hard work, but otherwise simple as pie. It's a violin plot. It's a box plot with the depth of the width of the distribution outlined. For AITC, not a significant difference. It's not interesting. For phenylatyl isothiocyanate, though, about 2-fold difference in favor of the endophytes. It looks like that there is this compound group. Otherwise, this is much more antifungal than the previous one. So this compound group has to do something with the fungal community. There is a group who was thinking about the role of the benzenic glucosinolates as the reasons why Brassicaceae plants are not mycorrhizal, because there are plants with mycorrhizae with glucosinolates, but none of those contain the benzene group, subgroup of GSLs. So perhaps this has to do something with the same thing, this. Okay. So what can be concluded from all of this? Lots of fungi have the ability to decompose these thioglycosides. Of course, they want the sugar, but they can also cope with the ITC that's generated, as it seems like. So there is nice tolerance. And I think we can conclude that there is some minor difference. The numbers of the fungi tested are low. Perhaps a bit more mining would bring on additional phenomena or additional viewpoints. The same. This also suggests that these compounds are very important in the assembly of the microbiome. That's not a surprise. So it's well known or well suspected. And it looks like that some fungi can simply bypass this chemical bond the plant's putting together against them. Perhaps you ask these questions. So this is nice, everything is nice, but you were working in a glass and disposable plastic, and it's a model of a model of a model. Okay? So how does this look outside in the field? Can we measure anything related to this in the field? And I'll show you what we did, and I think we can. We started with a horseradish accession maintenance site back in 2018. There were more than 20 accessions maintained there for, as a like a hobby of an old researcher, and we use these to generate chemical variability, to check them for chemistry, and then pick a subset in which chemistry fungus correlations can be studied. Okay, so 20 accessions does not mean that all of them are massively different for chemistry. So that's why we needed a subset. We selected 8 for detailed analysis, and this thing was run for 2 consecutive years, 18 and 19. We milled the samples, the horseradish roots, after surface sterilization with cryogenic grinding, which meant we can use the same powder, the very same powder, for chemistry and metagenomic analysis of the fungal community. Okay. The chemistry was done by Orbitrap. We also did the glucosinolate determination for the 4 major ones. That is the targeted part. We did an untargeted metabolomics, which resulted in lots of features. I'll show you in a minute. And we used an Illumina sequencing that also had issues. Untargeted metabolomics, it's a bit more than 350 features. We call the chemical entities candidates features. These passed all the filters I mentioned, and then we tried to annotate, putatively identify, approximately identify the ones the most interesting. What have we found? If I were using only targeted metabolomics, I would never have bought glucosyl 1-hydroxy-indole 3-carboxylic acid, or this one, but these are relevant. So the Arabidopsis literature is full of these I3CA compounds for fungal defense, and the hydroxylated versions and the thiocystin and the CTC. So these are relevant. We could we could assess the relative abundance of the samples for these, as well as phospholipids, peptides, amino acid derivatives. We also found flavonoids. I wasn't expecting them either, but they were there. Quinic acid, ferulin, caffeic acid derivatives, so miscellaneous phenolics, and of course the good old glucosinolates. How was our chemical variability? This line in the middle separates the 2 years, and the color denotes each accession. At that point, we were thinking of accessions. We wanted to pool the data along year and accession. We dismissed this idea later because it appeared a better approach for model building. But anyway, you see that the flavonoids have a quite high year-to-year and within-year variability as well. For some compounds that pass the quality control, there's nothing. So we won't be able to build any model of this. It's just noise. It's perhaps a biosynthetic building block, constantly expressed enzymes put it together, and that's it. While other compounds might show some variability. Phospholipids also showed interesting patterns. Of these 10 accessions, we selected 8 that covered most of the chemical variability of the samples. So, like checking this compound, these two look equivalent. So one tick goes for G and H. Some of it can be discarded from the fungal mining. This is at the extreme, so this is kept. And we check this manually for lots of compounds of interest. Indole derivatives, the same, also some variability, some year-to-year variability. Okay. DNA. We wanted to assess the fungal community by selective amplification of the fungal ITS region from a plant. Well, this is not that straightforward as it turned out, because the classical primers that many use amplify plant DNA. This results in 99% of our reads is plant DNA. So we had to redevelop some ITS primers suitable for horseradish, and it turned out that our sterile in vitro horseradish plantlet still contains some endophytes. So there are some weak bands, but these field samples were promising. So we don't have a massive band that's sequenced and turns out to be all plants, but so we use the custom primer. That's the bottom line. We've come to the interesting part: who's inside? What is the fungal composition? It's not well visible, so I'll show you. The first 8 samples, the first 8 columns, is the soil in which we raise the crop. It has no significant year-to-year variability, which is very good, because we might say that perhaps the plant chemistry will be dominating the plant endophytic community. It will be influential, as the pool of fungi to select from did not change. So this is important. The second one is some sort of something completely else, like control. It's carrot. It's a Daucus scrotum in the same plot. Okay, apparently different, but no weird rainbow colors, because it's not that dominated by a single fungal clone. The rest is horseradish, and from this point to the right, these are other accessions, and this is horseradish from a completely different site. I won't talk about this. This other site, we are preparing a large-scale similar study from it, but we just wanted to see whether it's whether we are seeing a completely different pattern there. The answer is no. So it's kilometers apart, different soils, and the pattern is more or less the same. This looks like, I forgot to mention, the color is proportional to the relative abundance of the fungus. So this purple, blue squares denote samples that are overly dominated by a single fungal clone, or an ITS and an ASV. What have we found? These are the most dominant: Lactosphaerella, Tanetophorus, some unidentified ASV from Brassicaceae, Podospora, etc. There are a few that were explicitly described as Brassicaceae endophytes, so these are underlined now. And there are others that were described as endophytes of other plants. Okay, so what might this all mean? It looks like that soil is so much different from plants, regardless of being a carrot or a horseradish, that the plant niche is a very strong filter. So many of the fungi cannot just walk in. We knew that, but it's also apparently it's nicely visible. This is more problematic: sampling depth and dominance. How much sample do you need from a single plot to cover the whole fungal community? If I had only this set, is it representative to all of this? These fungi would never have been dominant if I only had these samples. So I don't know how it is in other species, but this sounds scary to me. That you need lots of replicates if you want to truly assess the endophytes of any real site. Okay. First, we started with principal component analysis, etc. Everybody loves it. Nice plots, everything separated, reds apart from blue ones. But discussing them is problematic. Therefore, we went for row sperm correlations. So each fungal feature's abundance was checked for correlation against each chemical feature. This results in 15,000 row correlation tests, and this amount of statistical tests require the adjustment of p-values to readjust the false discovery rate. But anyway, we found very nice results. This color is proportional to sperm correlation. White is 0, so the pale colors are not meaningful correlations. But intensive blue and reddish ones show that something is there. It's nice. It looks like there are some clusters of fungi that are affected by clusters of metabolites, but there are also freelancers that do not join any cluster. This is very important because we are checking one by one. And you might as well say that you are not testing the correlation against a simple compound, but the correlation against this group. But as there are freelancers, I think we're fine with this approach. Okay, this is the p-values. the adjusted p-values, many have passed. Again, is the sporadic guys. We think it's statistically okay to talk about these. Let's move to something you can read. This is normal font size. Which fungi are affected? If we check the effect size, which is the fungal abundance difference, so Q1 minus Q4 par SD. So the fungal abundance of the top quarter of samples regarding that chemistry, regarding that feature, and the bottom quarter of that of the samples for that chemistry, and divided by the standard deviation for fungal abundance, we get the effect size. It is similar to the p-value metric. It's like, you know, the physicians always talk about how much sigmas they obtained. This is the sigma, how much sigma the difference is. This is only the statistically significant differences. It looks like that many fungi are more than one, perhaps 1.5 standard deviations apart regarding the chemistry. We see Cetophoma, Monosporascus, Fusarium, some Agaromycetes, some Agaricus perhaps. This is the genus level of the fungal data set. We see nice effect sizes, and these are in fact well affected. At an order level, we can aggregate these fungi. The thing is more or less the same. Important is that this accounts for a third of all reads. The third of all fungal ITS that was sequenced in the data. It's quite a high amount, not extremely high, but quite a high amount. What can be the rest? Why are they not affected? Perhaps they are affected by something that could be measured on the proteome level, or perhaps there is always something that it's luck, chance. So the first one gets there is becoming the dominant, and there's not going to be any meaningful correlation if he's the dominant. Here comes the interesting part. The major glucosinase doesn't seem to matter. They're not inside this selection of statistical significance. Only one minor putatively identified glucosinolate made it, who had meaningful correlation with some fungi. What are we seeing here? We're seeing other aromatic compounds are unidentified. We are seeing peptides being massively there. Flavonoid glycosides and other glycosides seem to be quite strong. How can we interpret these correlations? Because these are just correlations. We should not mistake them for causative relationships. If we see a positive correlation, a logical background might be some positive feedback loop. It's like the fungus tries to enter the plant responds by increased biosynthesis, and this equilibrates at a point which a fungus can't tolerate, and the plant maintains its biosynthesis up there. Okay? Sounds nice. This logical, as far as we have some compound that have evidence for being antifungal. But you can also get a positive correlation for a primary metabolite monomer during an ongoing pathogenesis, and the fungus kills off all the cells. And I'm open for any additional ideas, because there has to be a lot of other explanations. Negative correlations may result from successful fungal arrest. Again, this is logical for an antifungal compound. Fungus tries to enter, plant increases the biosynthesis, and the fungus is eradicated. It's eliminated from the plant. I think that might look like a negative correlation. But again, if the fungus spreads and it uses up monomers from the apoplastic space, perhaps that could present a negative correlation as well. Again, any idea is welcome. I was very much surprised by this peptide thing because, like an algorithm spits out that this is a compound X is a putative peptide. Peptides are easy to sequence in MSMS. Okay, let's dig into it. And a closer look on the peptide class, and especially the top, has revealed this. It's a glucosinolate decomposition product. It's a glutathione adduct with the glucoiberine derived ITC. Wow. So it's the same story again, but it's a meaningful story, I think. So it's this chemical form that tells you that there is an ongoing war. Horseradish has lots of glucosinolates. Perhaps it cannot even increase its biosynthesis further. About a third of the total sulfur of the plant can be in glucosinolates. That's brutal. It's almost competing with households of metabolism. Okay, and apparently some downstream products still are there and are in fact responsible for the fungi. The others were also found. They fell out somewhere during the quality control test or the adduct removal. But in fact, I am very excited to dig into this product group in the very near future. What are the final conclusions? There is an interaction, that's for sure, between the fungi inside the plant and the plant metabolome. The culturome, as they call it, the endophytes you can get out and raise in petri dishes, have measurable adaptation, show measurable adaptation phenomenon. But the in vivo study also has lots of interesting clues. We are working on the same soil endophytic community, and the same species, yet chemistry shows that several constituents drive the endophytic community assembly together. And everyone seems to matter. Flavonoids, indole, glucosinolate decomposition products. Interestingly, in this case, the precursors do not seem to correlate, and that's a bit tough to fully understand at this point. But the bottom line is you should consider measuring some sort of downstream product when you consider either plant pathogenesis or other model with glucosinolates. And to use this, to get to these downstream products, you should use untargeted metabolomics. The future. This is that's my color, it's 8 pixels. It's a 4-hectare area with gray, slippery clay down there, yellow sand down here, and very lots of soil types. And we are going to do the same with this, with various different endophytic communities, I assume. And let's check what comes out of it. So it's 13 sampling points. We have bulk soils, rhizospheres, plant endophytes. We'll have plant chemistry. So this is the next step to the story. And I'm also trying to move to other Brassicaceae plants models, study these adducts, build synthetic fungal communities. We now have lots of fungi that are characterized by glucosinolate decomposition properties, and it would be interesting to mix those and use them as inoculant for any reason. And I'm open to any ideas and collaborations. Okay, so thank you for your attention. That was it.
Time for questions. Now we can go. Okay, Sándor, and in your opinion, then the chemistry of the plants in a determined area is more important to condition the microbiome than the soil or other environmental factors?
I cannot answer that question without looking in depth at other sites and other plants. But if I had the data on the soil of the on the field that has the same, it's a 4-hectare area with the same agrotechnology every year. It's a horseradish monoculture there. I could answer this, but I don't think it's. I don't think I can answer this now. It looks like that chemistry matters a lot, and you can think of it as something in the future to predict the community the plant would pick up, but I don't think I can rank habitat, soil, and chemistry and genotype right now. I have no data on the proteomic level, for example. That also might be a massive contributor. How much reactive oxygen species are generated in the plant cell surface, etc. So these are proteomic level issues. We could measure this, and so far that's it. This is part of the whole story, I think. The only thing I can say that it matters a lot, but it's not something that is the only one to be considered. I hope I answered your question. So.
Yes, other thing. I know Brassicaceae is a special kind of plant that, in fact, it doesn't have micro. But many of the fungal endophytes that colonize Brassica can colonize other plants, other species. And which could be the chemistry behind that? Because here you are talking about glucosinolates, but glucosinolates are only in Brassicas. Should be some similar compound or something completely different? I don't know.
This is also an interesting question. We have been seeing a mixture of ubiquitous and special and really specialized compounds here. I think the plant is evolved using compounds with very generic strategies against the pathogens and the colonizers themselves. So the flavonoids, the ITCs, and so on, actually all work like bleach. They react with biomolecules and disrupt generic functions. Therefore, I think in the far future, we won't be thinking of identical compounds or compound groups, but some chemical substructures that might interact with the surface of the fungi. So what I'm saying is perhaps if we had data on the target of each compound, how it behaves, what is its main target, why it is antifungal, then we might join all of the plants to one, and we might say that the carrot has a different set of compounds that in the end does the same as the ITC in brassicas. So evolution has prepared the plants to generate mixtures which are specific. They can unless the fungus has something special to deal with the compounds, they will be out of the plant. That's it. I think it's so this it will be merged to a large holistic understanding. It will take a few decades, but that's my current idea. Okay.
Congratulations for the presentation. I have a question in this sense when we follow. Do you think simulating could be the key glucosinolide or even the key metabolite in Brassicaceae and endophyte interaction, but even in mycorrhizal fungal interaction, possible interaction with these plants?
It is again. I do not want to repeat my previous answer. So, one brassica has obtained the ability to biosynthesize, or inherited the ability to biosynthesize one group, and others came up with other solutions. Actually, the main compound class is important, but the Arabidopsis studies show that perhaps the biotransformation, the downstream product, can be of more interest, and how it is biotransformed. For example, the penetration, the PAN2 mutants of Arabidopsis do not have the ability to generate ITCs at the fungal entry site. And regardless of the glucosinolate levels, it results in increased fungal colonization and pathogenesis against some strains. So I think that looking at the downstream products might give us a more complete view, because the precursor elicitation is only the very beginning. It is the prerequisite of fungal arrest, but it doesn't tell the whole story. There can be lots of elicitation for glucosinolates without fungal arrest. There are lots of examples in the literature when the sensitive brassica tries to eliminate the fungus, but it just doesn't work. And the tolerant strains, on the other hand, have show much smaller elicitation, but it works. Perhaps because the downstream pathway is much more stronger, and that is the causative agent for the fungal arrest. This is not evidenced. I'm just I'm just extrapolating what we found. So I'm guessing. Okay. So I don't think one could say that this class is key and the other one is not. It perhaps depends on the pathogen strategy. Is it biotrophic? Is it necrotrophic? So what's it going to do? The plant has a toolkit against everyone, and we do not know which glucosinolate is against whom. But it would be a very interesting research to find out. So what products are actually there? This suggests that it might be the ITCs. Many came up with additional glucosinolate, especially indolic downstream products. But if we compare the antifungal activity of these alternative downstream products compared to the ITCs, they're weak to ridiculous. So this raffinose acid and indomethylamine, indole 3 carbinol, indole 3 methanol, these are not as potent as the ITCs, and their penetration, because of their polarity, is also much worse. Oh, okay? Okay.
Thank you.
It's my pleasure.
So, no more questions? Okay, so thank you again, Sándor.
I thank you for the opportunity. Thank you.